Hello everybody, this is Ola. In this video, we will discuss the muscle physiology and we will begin with the very first part, structure of a skeletal muscle cell. So there are three types of muscle tissue, skeletal, smooth, and cardiac. The skeletal muscles are the only ones that are voluntary. Skeletal because they are attached to the skeleton, to bone. The smooth and cardiac muscles are involuntary. Both the skeletal and cardiac muscles under the microscope are striated, but the smooth muscle cell is spindle shaped like this. It has narrow ends. Our topic anyway is about the skeletal muscle. So each cell of a skeletal muscle that we call also fiber or cell are the same, has its cell membrane like any cell in the body. In case of the skeletal muscle, we call that cell membrane sarcolemma. As the neurons, the muscle sarcolemma is polarized. So it has negative charge from the inner side and it is positively charged from the outer side. The sarcolemma has invaginations going deeply in the cell that we call T-tubules. They go deeply through the cytoplasm that we call sarcoplasm in case of skeletal muscle cell. So here is the sarcolemma and here are the T-tubules invaginations from the skeletal muscle cell membrane. They are invaginated this way to be attached or close to the terminal cisternae from the sarcoplasmic reticulum. The sarcoplasmic reticulum is the structure inside our skeletal muscle cells that stores calcium. So when the action potential goes along the sarcolemma, it goes here through the T-tubules. This will trigger calcium release from its storage, which is the sarcoplasmic reticulum. So here in the middle, this is the T-tubule. On each side, we have a terminal cisterna from the sarcoplasmic reticulum. So once the action potential goes here along the T-tubule, it's going to stimulate opening of voltage-gated calcium channels, and the calcium is going to be released from its storage place to be free in the sarcoplasm. And calcium is important when it comes to skeletal muscle contraction that we will discuss later. Every skeletal muscle cell has multiple myofibrils. A myofibril is cylindrical structure that extends the entire length of the muscle fiber. So if this is the muscle cell, it has multiple myofibrils, multiple cylinders inside. So here is one muscle cell, and at this cross section you can see myofibrils. Each myofibril consists of smaller units that we call sarcomeres. The sarcomere is the functional unit of the skeletal muscle. At this level of the sarcomere, muscle contraction happens. So if you look at this myofibril, you will see multiple sarcomers. So this is one, this is another one, and a third one, and so on. Each sarcomer has two types of filaments. Thin ones that we call actin, and thick ones that we call myosin. Why the skeletal muscle is striated under the microscope? The striations are due to the presence of the myofilaments, myosin, and actin. So here is a one sarcomere, and the sarcomere is in the myofibril. It has multiple sarcomeres. Each myofibril has multiple sarcomeres. Each sarcomere goes from a Z disc to another Z disc. So here you will find another sarcomere and so on. Let's focus on one sarcomere. In the middle, the whole length of the myosin, which is the thick filaments, we call this is A band. So A band is the whole length of the myosin, and here we have some actin overlapping included here in that A band. The I band is found at the side. Each side of the sarcomere has an I band, and the I band has only actin. So there is an A band in the middle, which is the whole length of the myosin with some overlapping actin. On the sides, there are I bands. They have only actin. The H zone here in the middle is the part that has only myosin with no actin overlapping. We call that H zone. In the middle of the H zone, we call that M line. The thick filaments, myosin, they are in the middle of the sarcomer. They make the A band. Each thick filament, which is myosin, has two globular heads. One is called the actin binding site. So this is actin binding site. It is attached here to actin, and it's attracting actin to move to the center of the sarcomere during skeletal muscle contraction. The other head is the ATP binding head, and here is where ATP is attached, which is the source of energy for muscle cell contraction. How about the thin myofilaments, the actin ones? So this is one actin, and the actin is covered with a protein that we call tropomyosin, this pink one. What keeps tropomyosin in place is troponin, another protein. So during rest, actin is covered and protected by the tropomyosin, and troponin keeps that tropomyosin in place. It's like a lock. It's locking tropomyosin from moving away from the sites on actin that are specific to for myosin to attach to. So what happens during skeletal muscle contraction is that calcium ions, they are like a key for that lock troponin. They unlock troponin, then tropomyosin moves away. Now myosin becomes exposed. So now actin becomes exposed. So now the myosin head will be able to attach to actin and grab this to the center of the sarcomere, and this happens during skeletal muscle contraction. This leads to shortening of the sarcomere and so shortening of the skeletal muscle cell. Now with some review questions. Place the organizational level of muscle tissue in order, beginning with the entire muscle. So number one is going to be the entire muscle, and each muscle has groups of muscle cells that we call fascicles. So each fascicle has multiple muscle fibers, and then each muscle fiber has many myofibrils, and then each myofibril has sarcomeres. Each sarcomere has myofilaments, the thin actin and the thick myosin. Okay, so myofibrils are composed of protein filaments that make the sarcomere. The myofilaments are the actin and myosin, and they make the striations observed in skeletal muscle cells under the microscope. What is the component of the muscle cell or fiber that quickly transports a muscle impulse from the sarcolemma throughout the entire muscle fiber? It's the invaginations, which we call T tubules. The protein of thin filaments, the proteins of thin filaments are the tropomyosin covers actin, the thin filament, and then the troponin keeps tropomyosin in place to protect actin during rest. What do you call the muscle cell membrane? We call that sarcolemma. And there are T tubules. They are invaginations of the sarcolemma. What are the receptors on the muscle cells? They are the nicotinic receptors, and we discussed that previously. And the neurotransmitter stimulating them is acetylcholine. So when the action potential goes along the sarcolemma, this causes calcium ions to diffuse from their storage place that we call sarcoplasmic reticulum, and then they diffuse into the sarcoplasm, which is the cytoplasm of the skeletal muscle cell.